Thank you so much, guys. Thank you, Bruce. That's always, having an introductory, a presentation from Bruce is one of the highlights of everybody's career, I have to say. Thanks for being here, and thanks for organizing this. This is truly a touching moment for me. Don't expect a well-organized talk because, well, I also had to write a couple of grants. Bruce, you told me that I was not gonna write any more grants. Okay, between one thing now I got the memo, yes. It was really, I think it's been really very rewarding to take time and zoom out and think a little bit about the path, right? And the voyage, that's what the word I used there. Because it's really, getting to this point, it's really not about one person, and it's not false modesty, it's really, it takes a village, it's true. And I've been blessed to be in a very good village. You'll hear probably a lot about luck and fortune, which is, it's unavoidable, it plays an important role. And certainly I've been an incredibly lucky person from a personal standpoint and professional standpoint. So this is gonna be all a big thank you to all you guys and my family who are there. So try to not embarrass me, okay? I told them I'm a very smart person. Sometimes when I say something completely controversial, do you know I'm a full professor at Harvard? Okay, so I was trying to think very hard about a very original way of starting this. I'm gonna just start from where I'm from. I actually don't know the guys. I think it's Patrick from SpongeBob. I don't watch SpongeBob, but it was a meme that I found. I was born in this place here. So if you guys can spell Nushina Gicani's last name, you will not have trouble here. This place is called Vizzolo Predabissi. It's part of the metropolitan area of Milan, and it's in the north of Italy. And here you probably see a few familiar landmarks. This is the famous Duomo, which is the main cathedral. It's on top of it, there is a famous golden Madonna, La Madonnina. Our president would be very excited about the fact that it's all gold. If you go here, this is the Galleria Vittorio Emanuele. This is how it looks like, inside when they light it up. It's really beautiful, beautiful place. This is one of the most famous, maybe the most famous opera theater in the world, which is called La Scala. And for those of you who follow the real football, this is La Scala del Calcio. This is San Siro, okay, where AC Milan and Inter plays. I'm actually supporting Juventus, but that doesn't matter. This is actually something a little more local. So Vizzolo is actually about 10 miles south of the city limits, and this is actually it's a 1,000-year-old church, local church from my town. This is where my wife Nazma and I get married. So I just wanted to show a little bit also some of the local beauties. So in Milan, I did my undergrad, and this is really at this university. The university is a private university called Università Vita Salute San Raffaele. It's another mouthful, right? It's a small university, but it's very strong in terms of imaging, right? So what I probably should have mentioned is that I actually studied for 2 years in the city of Parma, and this is actually where I attended the lessons by Giacomo Rizzolatti, who is the inventor, the discoverer of mirror neurons. That's where I got really excited about the brain. But really, when I started really getting excited about imaging the brain was when I moved to this university, and I had the opportunity really to study on work on a very interesting project. So back when I was an undergrad in Italy, you usually have 4 or 5 years of undergrad. So it essentially was the equivalent of a bachelor plus master, right? So now things are changing, are more similar to here, but that's what I had to do, 5 years. And the last 1.5 or 2 years, typically you would do a real research study. And I had the opportunity to study, to do a brain imaging study in the laboratory of Daniela Perani. So in this study, I actually had an incredible fortune to study in a very interesting patient population. These are patients that had hand amputation, but then receive a hand transplant. So what you're seeing here, this is a picture I took of a patient who, I don't know if you can kind of see, but here, his actual arm ended up here, and that this was the area of the allograft, so the hand transplant from a dead donor. And as I meet these people, you can actually see how they would, over time, gain in more and more function. And this was an incredible thing to see. I was working together with a team that including Marco Lanzetta, who was involved in the first hand transplant in the world. And so we would image them before and after with fMRI. And I apologize for the free software folks. I was using Brain Voyager. This was 2001. I promise I know better. I know better now. So you can see here, these are the activation in response to the opening and closing of the transplanted hand. And sorry, it's in Italian, it's small, so it's really worse. But so at 3 months post surgery, which is yellow and red, and then at 5 months post-surgery is in green, you can see that, first of all, at 3 months, you can see already the activation of the hand area. That's incredible to me. And then you can see also shrink it a little bit. We think we kind of reflect some kind of maybe a less effort in the given the motor recovery, et cetera. But also what was interesting to see is what happened to the somatosensory representation of the mouth. So the patient would be asked to actually open across their mouth in the scanner, which probably gave a lot of artifacts in our images, which is why we never published this. But most importantly, allow us actually to map the somatosensory representation of the mouth. So if you look at the right hemisphere, which is contralateral to the non-amputated side, you can see whether pre-surgery, which is in yellow, or 3 or 5 months post-surgery, which is in blue. And I don't know where my pointer went. Yeah, you can see the representation was pretty stable, okay? But on the other side, which is contralateral to the transplanted hand, you can kind of visually see, the ventral shift from yellow to red to blue, which kind of reflects somatosensory remodeling. Now, unfortunately, for technical issue, we never got to publish this data. I think it's a shame, and I wish we could. But this is what we got to do. And then, I was totally hooked on brain imaging. I wanted to do something about brain imaging. And where do you go? Well, and what kind of field you choose? Well, one thing that I was very interested in, it was some aspect of neuroscience that would have clear clinical application. That's something that kind of drive me. And there was an opportunity really to go to Montreal, McGill University, and to study under the mentorship of Kathy Bushnell. Pioneer in pain imaging. Later on, she became scientific director at NCCIH, and then president of ISB, a very accomplished scientist. And I was really, I think I was the last graduate student who she mentored from the beginning till the end. I think I have this distinction, hopefully. But it was great being with her. And it was great to be at the Center for Research on Pain, McGill Center for Research on Pain, that eventually got renamed Alan Edwards Center for Research on Pain, because that was really, and that still is, a mecca in the field of pain, right? You have people like Jeff Mogul, Fernando Cervero, Gary Bennett. If you're not in the pain field, maybe you don't recognize this name, but in the pain field, these are really royalty, right? And sitting in the room with them and being exposed to all these incredible thinkers and innovators really eventually resonated in all I've done, to be honest with you. This is the beautiful Montreal. If anybody has been there, Montreal? How about in the winter? Okay, so this is what it looks like in the winter. So those of you who have been to Montreal in the winter will know, the elation of the beautiful, city, even when it's very cold. It's incredible. It's very vibrant, very culturally rich and enriching. But you must have experienced the rush of incredible, incontainable frustration when you start shoveling the snow for 20 minutes and then it's not your car. It has happened to me. And the other thing, I'm not joking, I don't have particularly sensitive teeth, but if you're walking and there is a lot of wind, you cannot smile. Your teeth start hurting. This was Montreal. But then I decided to move to Boston, which is better. Well, it's not like this anymore. I feel like it has not snowed as much, but used to be. Of course, I moved here. Why? Well, because, we got, of course, this is another mecca, a mecca of brain imaging. And I had the opportunity to be mentored by not one, not two, not three, but 4 postdoctoral mentors. Apparently, I needed extra help. And, Ajay Wasan, I think he might be online. I don't know. He was asking for the Zoom link. He's at Pitt now. It's vice chair for pain research in the Department of Anesthesia there. Of course, a wonderful Randy and Vitali, also wonderful, and the equally wonderful Rob Edwards. So these four to me, so so they are like I've always referred to my mentorship team as almost like a multi-headed creature, but very in a very affectionate way because they've always been incredible to me. Sometimes when you go to these guys know about it. You go to mama and mama says no, you go to daddy, right? And I was doing the thing, but I had four parents to go to. And each of them really was incredibly supportive and allowed me really to grow as a person and intellectually living this time and space to think, to be creative. They provided the right guidance. So you hear a lot about talking about me talking about luck, and this is certainly one of the luck of my life, having these people. And so what were the projects that I was doing when I came here as a postdoctoral fellow? So we did a lot of work to try to understand how we can image clinical pain. Let me just explain a little bit. So how do most people try to image brain correlates of pain perception? You usually take somebody, you put them in a scanner, you give them some exogenous pain stimuli, and then you see what happened, right? With bold fMRI, typically, right? Now, this works very well if what you're interested in is acute pain, right? You can turn it on and off many times, many repetition. The stimuli can be short enough that, the right, temporal frequency that, hit the right, sweet spot of sensitive bold fMRI, etc. If what you're interested in, though, is clinical pain, well, clinical pain is a little problematic, right? So to image, right? Usually, you could potentially exacerbate it, increase it, but usually you don't turn it off. Certainly you cannot turn it on and off many times, right? So that's why we were trying to figure out ways to image using a different technique. And we explored the use of this technique called arterial spin labeling, ASL, that many of you will know about, and one experiment, and this was really, I think it was Ajay's intuition to start looking at ASL. So I'm very grateful. And he put me on the task, say, hey, you figure out how to make it work. And I think we made, because we took patients with lumbar radiculopathy, essentially back pain and sciatica, and we imaged them with ASL before and after, exacerbating their pain. It was for a good cause. I know it sounds bad, but for a good cause. So Ajay would use a contraption to kind of increase, raise the leg, basically performing some kind of maneuvers that are commonly done clinically, right, to diagnose. It's not like sadism, right? And then we image them the patient before and after this procedure. And by doing that, we're able to show that, yes, we are able to detect increase in the regional cerebral blood flow using ASL. And then I decided, well, usually ASL, how do people use ASL? They take all this data for maybe 6 minutes, 10 minutes, and then they collapse them and create a single map, right? There's a lot of averaging, and then you do some math, and you get to a regional cerebral blood flow map in absolute quantification in milliliter per annum grafts of tissue, right? I thought, well, maybe why don't we avoid throwing away all the important temporal information, right? We don't just collapse everything. We try to see if we can look for patterns of functional connectivity. Can we use ASL to see how different brain regions work? And by doing that, I think we actually got something quite exciting. This is actually one of my favorite papers that I've ever first authored. And so we were able to show that a specific pattern of functional connectivity, which is the connectivity between the insula and the femoral network, show proportional strength that is proportional to the clinical pain. So this is an important thing, right? So it's very strongly correlated to the self-reported pain. And we started a baseline. So even before doing anything, before doing any maneuvers, when we asked the patients, how do you feel? How bad is your pain? We were using a scale between 0 and 20 in that case, right? Well, we found that even at baseline, the strength of functional connectivity between insula and the femoral network would be nicely correlated with the self-report. But also, when we perform this exacerbation, that using those clinical maneuvers I described before, we found that the more their pain increased, the more the insula DMN connectivity also increased. Why was that exciting? Well, I was excited because finding anything that correlates to clinical pain is exciting. As you know, there is no objective biomarker. There's no blood test that tells you have pain, how much pain you have, right? But what was really exciting, and this was beautifully paralleling the discovery that Vitali, our wonderful Vitali, did first in 2010. Vitali, is that 2010? The first in patients with fibromyalgia, he has observed the same exact phenomenon a few years before in patients with fibromyalgia. And by showing that this actually can be replicated in different patient population, this actually suggests that these patterns might be an global marker of pain pain severity, which I think is actually one of the few biomarkers that I think is still holding true over, in the year. So I'm really excited to work with that. Okay, you guys, we're just the first 2 slides out of 59. How are you guys doing? Keep on going, or okay? So beginning of third year of my postdoctoral fellowship, and I started thinking, well, let me think about something that would allow me to create some kind of a niche for myself. I wanna, I like this work, but I really want to do something that is really, really, truly 100%. I don't want to say mine because nothing is mine, just like but I want to do something new, a little bit new. And I just this idea, hey, why don't we image neuroinflammation in humans with chronic pain? Did I have this idea first in the history? No. The people like the wonderful folks at McGill that I had the opportunity to meet and work with had, and many more, had demonstrated if you take an animal and that animal is injured, if you look at the spinal cord, but actually if you look at even up to the brain, you'll see that these cells that are the glial cells, that are capable of detecting this challenge to hemostasis, and they respond to it, but undergoing a series of cellular and molecular responses that we call glial activation, neuroinflammation. There are different terms, right? And this is exciting because if you block that pharmacologically or otherwise, well, the pain behavior are prevented, are reversed, etc. So incredible. Can we use modulation of immune responses to treat human pain? Think about it. We don't do that now, right? But could we do it? And well, the problem is that we didn't know to what extent neuroinflammation did happen in humans with chronic pain, certainly not until very recently. And so we started thinking about, can we do that? And so I went to this gentleman here, Professor Bruce Rosen, and I think I talked to you, I can't remember when it was, like sometimes in 2000. Anyway, doesn't matter. Let's not go there to the date because it's going to be wrong. And Bruce said, well, I think it's actually a good idea. I saw a little spark in your eye. So that was I'm going to introduce you to 2 young faculty at the center who know about PET and PET MRI. And one was Jacob Hooker, and the other one is Cyprian, who's over there. Jacob recently sent me this email that I wrote to him as a postdoctoral fellow, dated 2010, right? Sunday, November 21. And this was the email in which I was saying, hey, I would like to do some glial imaging in chronic pain. You think we could do it? And this was a slight endorsed moment for me, because think about it, if I had not gone to Bruce and got the courage to talk to this big center director, despite the fact that Bruce, you're of course incredibly approachable, right? It takes it's not easy just to walk and take an appointment with you. But what if I had not done that? What if I had not followed up with an email to Jacob and Cyprian, similar email I sent to you, Cyprian, as well? I guess the message here is like just go for it. I guess if I can teach anything to anybody, is just go for it. And I don't know if I like to think about myself as Gwyneth Paltrow, but so there you go. Now we got the multi-headed Hydra actually got 3. I forgot if it requires cutting one head to get 2 more that grows. Do you guys remember? I don't know. Anyway, we don't want to go there. Okay, so how do we fund this? I mean, great idea, but how do we fund this? So I submitted my K-99 June 2011, not discussed. Why? I think, they were concerned about my productivity. It wasn't, a lot of our stuff really was in the, we were cooking, the papers were ready to be submitted, but there was not a lot in the postdoctoral years, and they wanted to see that. Plus, honestly, It was not the best grant I've ever written, to be honest with you. The mentoring plan, which I was supposed to kind of, it didn't, yeah. So it didn't quite work out. Okay, sorry. I was learning, but this was a great learning opportunity for me. Resubmitted, not discussed again. So I guess other second message for you guys, especially in the crowd who are writing grants, don't get discouraged, right? Now, I wanted to point out a few interesting comments I got. So one weakness, sorry, Bruce, I know that I'm not supposed to roast you, but Dr. Rosen has listed 20 papers in his biosketch and a 2009 paper as most recent in section C of his biosketch, a PubMed search with Rosen, BR, and Boston. Yale did only one paper. All of this to say that it's Bruce's fault that I didn't get the grant. Okay, then we fixed the biosketch and, it became, okay, published actively, very strong research funding, etc. Okay, then, sorry, Bruce, what were you saying? You remember that? And then, I try my luck again because I'm a pain and, that's why I'm attracted to pain. And together with Jacob, under whose name the grant went, we applied for a Harvard Catalyst, was 50K, it sounds, good money, but it's not. And in reality, again, it's Bruce's, sponsorship of this whole venture is allowed to kick us out. But this was great. First grant, it felt so good, right? So I'm not going to go into details about what TSPO is. I'm happy to talk to you about 2012. We get the first PBR28 image. It was a moment that there wasn't a lot of fanfare, but I started thinking maybe this works. Maybe something. What if it's gonna work, right? So I remember when Jacob sent me this image or a similar image, I had to actually refish it from the back. Now, fast forward to today. I was asking Ellie, how many PBR20? Forget about Deprenyl, forget about it. How many PBR? And we have completed 820 PBR28 scans. Now, as I think about this, think about volume, but I'm thinking mostly about, the people, the hours that everybody put, all the patients, all the radio pharmacy, and the technicians, the postdoctoral fellow. It's really something. So that we did something like this together. Think about it. So I just want to say thank you for all the hard work. I don't want to get emotional. So what did we do with 820 PBR28 scans, right? We published an N equal 10 plus 9 paper. No, well, this was the first one. So this is the first paper that we actually published. For us, was quite foundational. It's really, put us on the map and let people think that what we were doing had some potential legs. So we started with chronic back pain and we imaged them with PBR28. You can immediately see how this is a median image. You can immediately see how there's a lot of signal in this part of the brain, which is the thalamus, compared to healthy controls. What was exciting to us was really looking at individual data, because in that data set, it's not, I don't know if it's something we have seen again, but there was such a beautiful separation between patient and control. Right now, you don't know that right now I have a bit of a neck pain. You know it only because I'm telling you. There's no way for you to know, right? All of this to say that how do we assess pain? Well, we ask people, right? Isn't it incredible? Here, we're able to tell who had back pain, who is a control, was demographically matched, but just by looking at these images. So that's why, despite the fact there were more co-authors that patients scanned, we felt it was kind of exciting. Okay. By the way, we replicated it, so it's incredible, etc. And let me show off a little bit. This was actually the top cited article of the 2015 or 2016 biennium for the journal Brain. So really, we're getting a lot of attention. And we also got the first grant, right? The first R01. And this is an image of my two favorite Dans. So Dan Michael Ellingson, who's now Professor at Oslo, and Dan Albright, my very first postdoctoral fellow, who's now a very established scientist in pharma. And this short here is me. We are in Vitali's office, which now is my office. Vitali was not there, and we snuck in. We snuck in Have I ever told you that, Vitaly, or no? Okay. Oh, did I put a phone in my mouth? So no, but okay, let me explain. We snuck in to steal some of the Norwegian aquavit that Dan Michael had brought it. So it's in a way it was his, right? So okay, so anyway, after this, again, this really opened the floodgates for us. We had many, many collaboration. We're able to show that this elevation in TSPO PET signal in the brain is not a back. We are not just lucky to have selected the single condition that shows it, but we saw it in many conditions: fibromyalgia, gulf for illness, migraine. And an important thing that you're seeing here, this opened my career to many collaboration. Eva Kosse from Karolinska Institute. Let me actually tell you one thing about Eva. I was actually I think we were at a conference in somewhere at a poster, and we were not initially collaborating. We found out at that post, in front of the poster, that, well, you know what, I'm doing a study, and now I'm doing a study in fibromyalgia. And she's telling me, oh, I'm doing a study fibromyalgia with PBR28 as well. So we were doing the same study, and all of a sudden we start saying, okay, let's keep in touch and see how things go. And then it was a little bit of a dance, right? There was a little bit of dance, and then she's very established. I'm a young faculty. I feel like, oh, this is kind of like my thing. I want to be the first. You're. I think, and I think I'm less silly than I used to be from that point. But in the end, we decided, listen, why do we do this? Why don't we put together the data and make this study that's immediately a multi-site trial, if you will, right? And we are glad that we did it because these effects were immediately replicated at Boston and Stockholm. We saw very similar effects, and this was an incredible stroke of luck, right? That's another sliding door moment, right? When both of us were at the same poster together. Um, Incredible collaboration with Kim Sullivan at Boston University. She's an expert in Gulf War illness. Incredible collaboration with Nushin, who now know how to spell her last name because I know Vitsalo Bradavis. And many more, right? So this was actually the really big part of the PPG that we actually got with Vitali and Bruce. So and of course, even beyond pain, right? So I was kind of interested in the angle of, in the neuroinflammation angle for pain. But if you think about it, it's a little weird, right? So it's much more easy to think about neuroinflammation when there is a neurodegenerative process, right? Anyway, this allowed me, because we were developing all these methods, etc. So we were fortunate to work with a lot of people. You can see Nazem Matassi, and this is Nicole and Katerina and her work in multiple sclerosis. She really has a very active program with many, many studies. Dana Rose's Huntington disease. This is Ludovica Brusaferri, who now is a faculty at London South Bank University, and she's done very interesting, a little scary work showing that even just being in the, witnessing the stressor of the pandemic seemed to have caused a little bit of inflammation in the brain, elevation of blood markers of inflammation, even brain aging. We don't want to go there. Let's pretend this never happened. Okay, I'm gonna keep on going. Then we get on, okay, so cool, we're seeing all this signal elevation in all this safer condition. By the way, it's not a specific, right? Sometimes I hear, okay, well, if you're seeing this in ALS and you see this in back pain, it's non-specific. Well, the specificity is the spatial distribution of the signal, right? So we are actually toying and trying to develop the idea that maybe we might be able to identify a neuroinflammatory signature that could be really of chronic pain conditions, right? It could be used for diagnostics and other reasons. Then we started saying, okay, does this signal move with treatment? So we start working in a condition that should be amenable to that, which is knee osteoarthritis. We studied patients with knee osteoarthritis before and after total knee replacement. And voila, we see that at least some of the signal in the thalamus and some basal ganglia actually normalizes after total knee replacement. Cool. This suggests that maybe this can be used as a potential biomarker for treatment effect. This is also one of the studies that allowed me to establish one of my favorite collaborations, which is Mohammed Jaraya, who is, where are you? Okay, he's a wonderful musculoskeletal radiologist. And then we decided, okay, well, let's look at even beyond the brain. What about the spinal cord? Right now, if I go back to Montreal and I talk to my colleagues at McGill Center for Research on Pain, they will tell me, of course, I mean, glial activation, that's what we see. We see it in the spinal cord. We just rarely go to the brain, right? Of course, us imaging people, you start with the brain, it's a little easier, moves a little bit less, right? It's a little bigger, right? But so we decided, okay, why don't we actually take a look and see if we can see anything at the level of the spinal cord? And this is a collaboration with Yi Zhang, wonderful clinician, now is the MGB division chief of pain medicine. Whoops. And this is Norman Kettner, another incredible mentor. He's a chair of radiology of Logan University and a long-standing collaborator of a few of us, Vitaly Bruce and myself. And we were able to see elevation in the spinal cord. We were able to see even at the level of the neuroforamen, which is the part of the spine where you have the nerve roots and where your physician, or maybe Mohammed, places the needles to do an epidural steroid injection, right? So we're actually started asking the question, does that hotspot predict who will respond to the epidural steroid injection from those who don't? And, tiny study, but, the results were kind of interesting. So these are the subjects that do benefit from epidural spinal injection. You can see a higher signal compared to those that don't. And of course, this opened the door for bigger, better power studies, and really many more studies to come. Then we went a little wild, right? So because up to now we were still in the central nervous system, well, plus the peripheral nervous system if you count the neuroforamen. But we wonder, okay, well, what about the periphery? Is TSPO really, everybody uses to study brain inflammation, but if you actually think a little bit about it, it's also expressed in peripheral immune cells. So can we then use it to image inflammation maybe in the entire body? Isn't it cool, right? The idea that maybe you can take a single patient, and I wish we had maybe some technology one day that allows to image the entire body. Okay, Chiprin, you know what I'm talking about. And so we say, okay, we were scanning patients with the arthritis. We're scanning 90 minutes their brain. And so how many half-life have passed? Several half-life, very little activity, but we move the field of view to the knee joints and say, are we able to see anything? And voila, this is an example. Of course, these are representative examples, but there are examples of healthy volunteers, patient with the unilateral knee osteoarthritis, patient with bilateral knee osteoarthritis. We saw clear elevations, proportional to pain, proportional to inflammatory markers, et cetera, et cetera. So this really, this to me opens the door to the possibility really to image an entire person, right? So inflammation throughout the body. We tend to be very siloed in what we do, right? So all I know is, brain, I've done brain, and Mohammed maybe, sorry for, I keep calling you, but Mohammed's focused on his knees because that's what we see all day. Maybe it's time to all start talking to each other and look at an entire person. So this is going to be probably one of the next chapters. And then you start looking at the entire person. Now you start making, not only confirming, yeah, okay, well, this is cool to see, but you can expect, right, that there is pathology in the knee of patient with osteoarthritis, right? But now we started finding also some interesting and completely unexpected and potentially, I think, important findings. For instance, the role of the bone marrow in the skull. And the first report was really done by Nushin in 2020. That's what we published. We published 2020. I thought it was before. Maybe came after. Okay. Anyway, we published sometimes around 1920, etc. And we found this interesting observation. Patients with migraine show this elevated TSPO PET signal at the level of the skull bone marrow and adjacent meninges, right? And fast forward, this is the collaboration then also with Vitaly and Bruce, etc. And we are replicating essentially the observation in a much larger number of subjects. You can see how it's actually proportional to the frequency of migraine. So it's not just an epiphenomenon, but seems to really have some clinical significance. And then we're bringing this to, this is obviously not just a migraine thing, because we're seeing this this is a large study. This is 125 patients with chronic pain. We have 88 back pain patients with knee pain. You can see these massive effects. And it's proportional to pain intensity. It's proportional to knee pain in patient with knee pain. It's proportional to back pain in patient with back pain. It's proportional to interference, anxiety, depression, sleep disturbance, really something important. And if you actually zoom out in 2023, this paper came out in Cell. This is not us. And they actually show that what we had described initially in migraine, it's not a migraine thing, can be also seen in Alzheimer's, tauopathies, stroke, multiple sclerosis, etc. So I think this is a very basic disease response that I'm actually quite excited about. And I think we're gonna really, I'm trying to look at, some of my favorite radiochemists, hoping that they can develop a tool to look at, yes, she's saying yes, okay. Okay, a little bit of self-promotion. We got a lot of, good coverage from different media outlets. Some were more exciting than others. We got, TikTok. We got somebody actually reported on TikTok. Actually, they saw, one day I will show you. They got like millions and millions of views reporting on the pandemic brain paper. It's incredible. We got featured Buzz, BuzzFeed Daily and all this stuff. And if you're wondering if this is Lady Gaga, yes, this is Lady Gaga. Why? Well, this is because I started getting interview requests when she was diagnosed with fibromyalgia. So I don't know. Yeah. And then, I have to say some of the like moments that I kind of was particularly exciting to me is when I found out that Nora Volkow and the director of NIDA and Francis Collins, former director of NIH, actually were presenting our slides. To me, it's like, I'm sure I'm not the only one here, but to me it was kind of, hey, this is going places. And then there's a moment in which I almost had a heart attack when actually Francis Collins invited me for a meeting. That was in 2000, I forgot, 17. And it was a meeting with about 30 people, of whom there was one Nobel Prize. There was people really at the top of their game. And I was there like, already I'm a diminutive stature, but all these giants, and it's, oh my gosh. And of course, I got invited, big talk, etc. Some talks were very attended. This was in the plenary session I gave at ISP 2024. There were probably 2,000 to 3,000 people. I was giving the talk for the people that you guys don't know. I was giving the talk to you guys. There was a few people in my lab, and I was so proud for them because I was saying, this is stuff that we did, right? It's incredible. And then, yeah, I also got other talks that are a little bit less attended. This is the this, hey, okay. This is the organizer of the so okay, so it was 7 p.m. on a Saturday night in the no, let me justify in the gastronomic capital of France, which is Lyon. So, this is not a bad turnout, okay? Then yes, I want to also celebrate my neuroanatomy for dummies. If you don't know, you can learn neuroanatomy by going to YouTube and Google neuroanatomy for dummies. We actually got 116,000 views, which is less than Zainab's cat get in a minute. So she does all anyway. But I love the comments, right? So I got God bless you, amazing. But this is the best one. He sounds like Cesar Millan. First of all, you guys know Cesar Millan is the dog whisperer. First of all, wrong accent, man. Anyway. And this one here. Okay. Did you guys know that I got interviewed by Larry King? Do you guys know? No, me neither, because it's not true. So, so one day there was this guy that calls my office and say, Dr. Loggia, I forgot, from Louisiana, whatever, rheumatologist, or, I'm reaching out because, I know that Larry King wants to interview me, and they sent me the media package, and I can see that, they interviewed you, like, upcoming show, we'll be taking you viewers there, Marco Loggia, instructor in radiology. And I said, that's weird, that never happened. Maybe that is why they're asking me to cover some of the production costs. So they are asking this guy pay some money to be featured in this Larry King show. And anyway, so if my name starts used for scamming people, I feel I made it, guys. And then there was this one. Then I'm gonna let you. I know you guys probably want to eat something, but so I wrote this editorial: chronic pain in opioid receptor availability, disentangling the molecular contribution, and the chicken or the egg dilemma. So you guys know what I was going for. So I was trying to figure out whether alteration in the expression of these receptors could be cause or an effect, right? And then for some reason, I got invited to contribute to poultry farming. So I love interdisciplinary research, but I had to pass on this one. Okay, so who know this guy? Do you guys know what it is? Yeah, Slash. Okay, 1993, my first concert. By the way, these kids at that age, oh my God, imagine sending them to see a Guns N' Roses concert. No, Nazma said no. So where do we go now? And I promise I'll let you go soon. I guess like a little sampling menu of what we are working on now. Too many things, honestly, and too many people to acknowledge. I'll really brief through it in case somebody gets excited. Just reach out to me. Why don't we do it this way? We are first of all exploring. We're doing more TSPO, right? Um, as much as I, also want to do other stuff, there is so many questions that still need to be answered, right? Um, we're focused on different conditions, such as this is a study we're doing together with Yahui Zhu, my former postdoctoral fellow, who now is an assistant professor at the University of Indiana. She's very much interested in AIMS, which is the um, aromatase inhibitor associated musculoskeletal syndrome. This is a kind of widespread musculoskeletal pain that is very debilitating, and it's very often a side effect of this medication for for breast cancer. Sometimes people had to stop their medication because they're in so much pain. We're trying to understand that type of pain. We're focusing on HIV. I try to understand HIV neuropathy, potential interaction with opioid use. This is a study with Eva and Shibani Mukherjee and many others. Ellie's writing a paper. You're writing a paper? She's laughing. Yes, she's working on writing the paper. Okay. We're with Thomas Chang, we're doing a study on orofacial pain. Ikem is doing an interesting, exciting study on the effect of trauma and neuroinflammation. And then we are also looking at TSPO as a potential marker of treatment response in many different clinical trial, mechanistic clinical trial with the spinal cord stimulation. This is with Brian Wenger and I think I have to say Brian Wenger and Roy Freeman. We have a study with Jody Gilman to study the effect of cannabidiol. We have a collaboration with American University, Kate Holton, to study the effect of low glutamate diet in veterans with Gulf War illness. We have, the aforementioned PPG study we're still working on together with Vitaly and Bruce and Ushin and many others, and Randy and many others, Ronald, studied the effect of transcutaneous vagus stimulation. We have this work also with Vitaly, try to understand the effect of carpal tunnel syndrome, et cetera, et cetera. And then we're moving towards the concept that was mentioned before, the idea of image the entire person, right? So the idea of not focusing on one body part, but really try to understand the entirety of the person in pain and using inflammation as our, trait, fil rouge, if you will. Now, and of course, these efforts will be rendered, achievable by this amazing technology we're about to get, which is the total body PET-CT. I'm really excited to use it as soon as possible. Okay, I know you guys are probably thinking, okay, TSPO, oh my gosh, is there anything else other than TSPO? This didn't land very well, this joke. I'm going to explain. He's disappointed. Okay, all right. Okay, are the kids laughing or no? Yeah, no one's laughing. They're probably embarrassed. They have to deal with so many dad jokes, guys. Anyway, so we are going beyond TSPO. So those of you, we're also focusing on different radioligands. For instance, we study veterans with Gulf War illness using this ligand that is supposed to tell us something about their astrocyte activity, AMAOB expression, which is L-deperanol. If you're just going to talk to me, we are using the Connectin 2.0 to try to understand the microstructure, try to see if we can detect the kind of microstructural alteration we're expecting histologically, the retraction of the processes, the increase in cellularity, the swelling of the cell body, etc. We expect to find inflammation using, advanced diffusion imaging, Then we're doing this work, and this is really led by Merbal Mohammedian using low field MRI. We're trying to see if we can actually bring imaging to the clinic. Can we detect in patients with chronic pain conditions, specifically knee osteoarthritis, the kind of alteration that can be used to, let's say, predict those who are gonna have adverse outcome, let's say, poor outcome to total knee replacement. And this is, of course, the work we're doing with my favorite bass player, who is Matt Rosen, and with Taylor Kimberly. Then I know that we got some work done in terms of where's Maggie, you know, that MID, yes. So we're looking at with both fMRI, you know, the reward processing. We can talk a little bit about it. We're doing some structural work. We are also doing a lot of work with spectroscopy and so forth and so forth. I'm almost done. And this is really the most, if you forget about everything I've showed you so far, that's okay. Let's focus just on this one. I want to say a huge heartfelt thank you to all the people that I really had the opportunity to work with, to mentor, to see grow. You know, of course, you know, our group went from very lean to, you know, pretty big at some point. Now probably going to get a little smaller again and maybe big again, or maybe disappear depending on NIH. But, you know, I have to say, again, there's a lot of luck involved, and I was really lucky to really work with you guys. I want to single out one individual. So Ellie, amazing. Everybody should have Ellie in their life. So she's really the glue from the lab, right? So you don't know how many times I hear people from, you know, the techs or the CTRU or oh my God, your group is so organized and they, you know, and they're so lovely, so incredible. And, you know, and a lot of it is really the culture. And Ellie has been really, you know, a cornerstone to this whole thing. So I just want to say thank you. Of course, I have to mention, I can, you know, 820 PBR, you know, the radiopharmacy team, which of course has evolved over time. They work so hard and you know, they really are the people that make us happen. Wait a minute. Yeah. Okay. Of course, sometimes we forget, but we are one of the reasons why we're so lucky to be at the Martinos Center is these people. I have to say, we are so lucky to work. I'm not even mention any of them because each of them are really a crucial role. And maybe I'll mention something. I'll mention Stacy because she's actually running a stealth operation. You know why? Because she was able to hide that chair from me for all this time. Like, so this was in the kitchen. Surprise, captain chair. And for those of you who don't know, so the Gardner, this is actually the place that makes the famous Harvard chair, right? Anyway, a big thank you to the, of course, center and radiology department leadership. Bruce, you've been an incredible mentor. You're, you know, the mentor that I want to be. Of course, Dr. Brink for the vision and you know, the support, and of course, Susie and Hope. We're really lucky to have you guys lead us and help us really do what we're doing. I should be remiss if I don't mention my second home, which is the Department of Anesthesia. And this is Sheun Akeju, who, when he's not being a fashion model, looks like a fashion model, is actually the MGB chair of anesthesiology, an incredibly accomplished and talented scientist, clinician, humble and you know, and a good friend. He's been really supportive. I mean, okay, now as chair of MGB, probably is not doing this, but he was the guy who come at, you know, whatever crazy hours in the morning to come and place an ALAN for us after he had not slept all night. Because, you know, this is stuff that I don't forget, right? So, you know, we need more people like him. And of course, many more people in the department. Ariel Mueller, administrative director, she's incredible. And then all the people that really also help us out, Anake and then Vavika Shah and Dan Sadawi Konevka, they're the people that help I actually set up the pipeline that brings all the provider that place A-lines here. So those of you who are doing A-line study, arterial line studies for PET, we have to thank these people. That they are the reason why we now, after 820, PET, the PVR scans. We can do it relatively well. I think we are. And of course, I'm going to end, I think, with the community. Martino Center is incredible, very vibrant. I really feel at home is here. I don't think there's another place that I could be as happy as I am here. So thank you so much. And maybe I'll end actually with the, talking about luck, I truly lucked out meeting these people. Well, making three, I met one and I made three together with. So Nazma, we met in Montreal. She was actually a research assistant in Kathy's lab where I did my PhD. It was kind of a little problematic there, but she swears she didn't like me. And I promise you, she told me multiple times. And I told her, I think you did like me. We had 3 kids, so you guys decide. This is beautiful Dolomites. If you've ever been to Lago di Braies, it's an incredible place. It's pure magic. I think the family is not complete if I don't show her. So these are the people that wake me up at night, and this is the person that wakes me up in the morning. I kind of messed up. Keep me up at night, and then wake me up in the morning. And Coco is wonderful, and she's actually the person that loves me the most. I'm not joking. A huge thank you, really from the bottom of my heart. I'm hoping I'm pretty sure I will have forgotten people, but thank you again.